I was diagnosed with FSH when in the early 1980s. I'm in the back, and I had a muscle biopsy, but I don't know if it still exists. Would it still exist if it was done at Children's Hospital in Boston?
It probably does exist. I just don't, I would have to go back and look carefully, go through pathology, because pathology keeps almost all those muscle biopsies.
Thank you.
Anyone else?
For FSHD2, is there a database of all the different mutations associated with that? Because I think with whole exome testing, where at least I have found a patient that had a heterozygous change and really doesn't have the phenotype for FSHD. So I was just wondering if there is a database and how would one access that?
Yeah, there is a database with all published mutations, and we will soon populate that with the new mutations that we found. It's the Leiden Open Variant Database, LOVD, and it's basically a mutation database for all disease genes that are available, and it's open source, so you can easily check that. Leiden Open Variant Database.
I have a question. So when a spontaneous mutation occurs that causes FSHD, does this always result in type 1 or infantile FSHD?
No, it can be a adult onset FSHD. I think the most predictive feature of whether it's early onset or adult onset is the repeat size at this point. So very small repeats typically are associated with the early onset cases, and the somewhat longer arrays are associated with the adult onset. But it can also be a new mutation in SMCHD1.
Because in the literature it says usually when there's a spontaneous mutation that can result in infantile. So I was just wondering if there was a correlation.
Well,
there.
I think the difference that we're seeing is that the cases with infantile FSHD are typically the more severe end of the disease spectrum, and so they do not establish families. So they remain isolated cases in the families, right? And if you have an adult onset, that's very often actually symptoms start after you have given birth to children. So, and then you can have already transmitted the disease to your children. So that's the difference that we are often seeing.
Hi. Some of the vocabulary terms on your slides occasionally stretched my vocabulary, and I wanted to, you brought up the word gene methylation. Can you define that in a broad sense about what methylation means in genetics as opposed to chemistry?
Sure, actually that was the last slide after my acknowledgments I pulled out because I thought no one would be interested. And I knew I'd be running long. So it's a modification on cytosine, which is one of the 4 DNA bases. The methylation is put at the 5 position by an enzyme, DNA methyltransferase. It's modification is in humans or invertebrates. It's only in a specific set of dinucleotide, a CG. And this, since it's put on by an enzyme, it's actually removed by an enzyme. There are DNA demethylases, so this mark can be added and removed without changing the DNA sequence. And essentially this is, but because it's in a CG dinucleotide, the way DNA works, CG on one strand will be copied as CG on the other strand going the other direction. You always get a symmetrical DNA methylation, so that mark is always copied every time the DNA replicates. The enzymes recognize this as to restore the DNA methylation mark, and so the mark gets continued to be passed on through generations unless it's removed from both strands. So that's kind of our classic epigenetic mark because it's heritable and it's dynamic and it's reversible. It kind of fits all the characteristics. But the machinery of the cell, there are proteins that specifically recognize a sequence if it's methylated. Methyl DNA binding proteins. There are some proteins that cannot recognize a DNA sequence if it's methylated. So there are actually these profound effects by these. This one little, methyl group is a CH3. Just kind of this little group added onto a cytosine actually has profound effects for how the nuclear machinery sees the DNA. If you think of the DNA as a platform for assembly of nuclear machines, methylated platform is different than unmethylated, and it's seen, recognized differently by the cellular machinery.
Hi, here I'm at the back. So Alexandra Belayev. I wanted to make a comment about the epigenetic story and asymmetry. So you might be right. In fact, nobody knows exactly, but you might be right that indeed epigenetic could contribute to the asymmetry. But if you think of the transgenic zebrafish that Dr. Kunkel has presented, he has made those fish by injecting messenger RNA. So there is no DNA methylation involved, and the animals are asymmetric. So epigenetics is probably not the only answer to asymmetry.
Well, but the readout of epigenetics will be a mosaic expression of DUX4, and I'd predict that in the zebrafish that every cell does not have the same amount of DUX4 after development, and that the asymmetry could be explained by you can correct me if I'm wrong, this is just one of the ways that it could be, in my mind, explained is that as the organism is developing, different levels of DUX4 in the different cellular compartments leads to an asymmetrically developed animal, which would be very similar to a person whose epigenetics is somewhat mosaic during development as well. So that would be how I'd address that.
Probably more complicated than that, because among the genes which are direct targets of DUX4, you have the PITX1 gene, onto which Yi-Wen Chen has been working a lot, and there are strange things in the literature about mutations in that gene that cause asymmetric phenotype also. So there might be something involved in the way those homeodomain transcription factors are working that we still have to discover. But it's anyway very
completely agree that there's a lot to be done, and it's going to be complex interactions between many mechanisms. I agree completely.
I know researchers are reluctant to answer this question, but when can we expect a treatment? And when it comes, will it lean more towards a cure or more towards just stopping the symptoms from developing further?
This is the hardest question always, but I think that tomorrow Dr. Van Engelen will show you that although this is not a pharmacological treatment, but by being aware of your lifestyle, you can already accomplish a lot. And again, this is not going to be a magic bullet, it's not going to solve all problems, but it will show a reduction in the disease burden. So while we are working on drugs and on druggable targets, and it's unpredictable how long that will take, but it will take less long than I would anticipate 5 years ago, there are already things that we can do to release somewhat of the disease burden in this disease. And you will hear about that tomorrow from Dr. Van Engelen.
First of all, I just want to thank all the participants. I feel somewhat intimidated by all the massive brain power assimilated in the front of the room there, and really almost hesitated to ask a question, but I'm going to ask it anyway. I did not present any symptoms until I was in my late 50s and was not diagnosed officially until I was 59. It's always been a curiosity to me why I exhibited no symptoms in my youth and right up past my middle adulthood, and wondered if anyone could offer any suggestion as to how this might have occurred or why. Or
well, we believe that there are modifiers of the disease severity, and that's one of the things we're trying to track down right now, as to why people like you develop symptoms very, very late in life, and she had showed none before that. And I think the only way we're going to get at those is to understand the genetics of it. as well as potentially the epigenetics.
And the epigenetics.
But I would answer the question that came earlier also about therapies. I think we're on the cusp of therapies for most of the muscular dystrophies, including, I think, FSH, but none of them are going to be curative. They are going to be slow progression. They're going to change disease course, I think, initially. And then, as time goes on, we may get more curative.
So I just want to, of course, put another plug in for epigenetics, though. But one of the things I didn't get into, we look at DNA methylation, but what Stephen Tapscott's lab showed very nicely is that most of your FSH, most of your muscle cells, if you're FSH affected, are not expressing DUX4. It's actually very few cells are expressing that, even though. So the epigenetics say that it's amenable. That's why I put down amenable and not necessarily on. And one of the things we've been looking at is the difference between FSH affected and asymptomatic. And what it looks like is that in affected individuals, those cells really want to express. They're on the cusp of expressing DUX4, and then periodically they express DUX4. And in the epigenetically altered, the asymptomatic, they're harder. They're not as poised. So we think of this as you're poised to express DUX4, which then causes problems with that muscle that's expressing it, and in asymptomatic you're less poised, and in healthy you're actually very stably repressed. So it's possible with age that essentially there can be some changes that come with epigenetically, where you go from a stage, even in FSH1, where you become more, going from the poised stage of DUX4 to expression to more cells expressing it, and that's when then you hit the threshold where you develop disease. And again, this is just a theory. We only have single time points where we look at muscle biopsies. What?
No, we didn't mean to interrupt you. Please finish your thought. I was just going to add on a clinical perspective to what you were saying.
Well, I was just going to say we only have time points, so this is just my theory of how we look at it and what we know from cell culture, but actually that's a good lead into Catherine can comment about clinically what you see.
Sure, just to add a clinical perspective to the question, that it is also possible that the same molecular and cellular insults are going on since birth with FSHD. We certainly think that's true with many of the other muscular dystrophies. And the physical manifestations of weakness is an accumulation of those insults. And so the physical manifestation is the replacement of muscle over time by fat and fibrosis. And so although you didn't have weakness until your 50s, I think you said, that the weakness now is because of a lack of muscle. And so that's on a very different level than the molecular issues that have been discussed. One of the really interesting things that's emerging, I think, in understanding FSH on a clinical perspective is how it may differ than other muscular dystrophies, in which we think that there's a slow progression and in which muscle is replaced by fat and fibrosis over time. But in FSH, these insults may happen very rapidly. That you may go from a normal muscle to a fibrotic muscle very, very quickly with only a small period of time. And many of you have expressed that you say, this year my biceps was picked off. And so that's something that is becoming apparent through MRI studies, some of which are supported by the FSH Society, and many of you have participated in those studies.
We have time for one or two more questions, or about 5 minutes behind schedule, so one or two more questions, and then we'll go to break.
I'm wondering, and you may have answered some of this in responding to the previous question, but is our understanding of the genetics and epigenetics still so recent that there simply has not been time to do studies that show changes in the epigenetics within one individual's lifetime and within the progression of the disease in one individual's lifetime? So, you know, although you say that epigenetics seem to be quite stable, are we uncertain of the fact that if you have, you know, person A who appears to show onset of symptoms quite late, or person B whose symptoms present with more severity earlier, is there a difference in how that person's genetic profile or epigenetic profile would look at periods of 5 years? Is this just still too new? And if it's still too new to have that kind of picture, is somebody starting to do that?
I think that's a very good question. I think the epigenetic marker that has been studied most widely in FSHD is DNA methylation. And that has always been considered for many, many years as a very stable marker that didn't really change over time. Some recent data show actually that also DNA methylation is dynamic, and there are regions in our genome that change with aging. The good thing about that is that you can test that because we have DNA from individuals already from 30, 40 years back, and we can redraw the DNA and look at it again. We have done a little bit of work on that already many years ago and didn't see much of a change in the DNA methylation. But again, this is only one of the modifications, and although it's a very good marker for the change that we see in the chromatin structure of D4C4, it's certainly not, or it's likely not the most necessary change, and perhaps even not directly correlated with DUX4 expression. So it might be that we need to look for other modifications and look at that more carefully in time or over time. And there we are really, we have the disadvantage that this realization about the epigenetic structure of the repeat is relatively recent, and so that it will require probably some more years to be able to compare that in different individuals from different age, and even preferably from the same individual with intervals of many years. Yeah.
Okay, I think we should go to break at this point. We're about 6 minutes behind schedule. I want to thank one round of applause to our presenters, please. Thank you. We should
Okay, just a couple housekeeping announcements. The bathrooms are open now, so it's the best round of applause I'll get all day. And I also wanted to let you know we have a professional photographer who has set up a photo studio, portable photo studio off to in that corner of the foyer. And there are sign-up sheets at the registration desk, and the objective of our photo project here is to create portraits of living with FSHD. The portrait, we want a portrait that tells a story to the public, because this is so important for raising awareness. And we have not nearly enough images to help us convey that story, the urgency of doing more research on FSHD, how this is impacting your life, and also that, you know, you're beautiful. So they're going to be wonderful art quality photos. There are limited number of time slots available from now through mid-afternoon. So if you're interested, please sign up for a time slot. And we also encourage families. It'd be wonderful to have family groups of family portraits. So thank you so much.